welcome to the Deep Dive, your shortcut to understanding complex topics, well, quickly, with fascinating facts and just enough humor to keep you engaged. Today, we're diving deep into the incredibly diverse, often unseen, and utterly crucial world of how we classify and, identify microorganisms. Our insights come straight from Chapter 10 of Microbiology, an introduction, the 13th edition, and our mission: to peel back the layers of this microbial universe, discover how scientists make sense of its vastness, and truly grasp why this invisible world is so fundamental to our health and the health of our planet. Here's something to get your brain buzzing right away. Did you know biologists estimate there could be as many as 100 million different living organisms on Earth? Yet we've only identified and classified, less than 10%? And what's truly remarkable is that we're living through what's being called the third golden age of microbiology, all thanks to dazzling new technologies. Get ready for some serious insights.
It's a remarkable time, yeah. A remarkable time to be studying microbes, absolutely. And what's truly profound about classifying them isn't just, giving them names. It's about uncovering their deep evolutionary relationships, tracking diseases with pinpoint accuracy, and ultimately appreciating the very foundation of life itself. Let's put a fine point on it, maybe. Consider this. How do we quickly identify the tiny, unseen culprit behind some mysterious pneumonia that affects both a patient and their pet, or even trace a nationwide foodborne outbreak back to its precise source? The answer is, well, they lie within the systems we're about to explore.
That's a perfect way to set the stage.
Yeah.
So, let's start at the beginning. The core concept, taxonomy. Simply put, taxonomy is the science of classifying living organisms. Think of it as the ultimate organizational system for life, establishing relationships, figuring out how different organisms vary, and giving scientists a universal language. No matter where they are in the world, it brings order to chaos, basically.
Right.
But it's not just about neat filing. This also ties into systematics, or phylogeny. That's the study of the evolutionary history of organisms, right?
Exactly. It shows us how every living thing, from the smallest bacterium to the largest whale, is connected through common ancestry. All life is truly intertwined.
Intertwined. And that intertwining is incredibly practical, you were saying.
Oh, absolutely. Let's go back to that pneumonia example. Imagine a 75-year-old cancer patient and their dog, both sick, similar symptoms. When the lab isolates bacteria from both, they find microbes that share certain chemical characteristics. They're what we call gram-negative, and they test positive for specific enzymes like oxidase and urease.
Okay.
These aren't just academic terms, they're like chemical fingerprints. By matching these specific fingerprints to known classified bacteria, doctors can quickly identify the pathogen and prescribe the right treatment, potentially saving lives.
So it's really frontline stuff.
It really is. This isn't just theory, it's life-saving work. And historically, our understanding of how to classify life, well, it's undergone some radical shifts. For centuries, we were kind of stuck with Aristotle's simple two kingdoms, plants and animals.
Right, that's all there was.
Then Carl Linnaeus came along in 1735 with his formal system. But he and those who followed, they really struggled to fit microscopic organisms, bacteria and fungi into that framework. Carl von Nageli even proposed putting bacteria and fungi in the plant kingdom.
With the plants.
Yeah, which, looking back with modern DNA insights, is pretty ironic. We now know fungi are actually evolutionarily closer to animals than to plants.
Wow, that is ironic. That's a fascinating historical detour. It really highlights how our understanding evolves. So from those early, sometimes misguided attempts, we saw a major leap forward. 1969, Robert H. Whittaker's five-kingdom system. This system finally gave prokaryotes organisms without a true nucleus their own kingdom, monera. Eukaryotes filled out the other four. But here's where it gets really interesting, and where the aha moment truly comes in. The ultimate game changer arrived with breakthroughs in molecular biology, specifically the ability to compare ribosomal rRNA sequences.
That's precisely right. That molecular approach, pioneered by Carl Linnaeus in 1978, it completely revolutionized our understanding. He proposed elevating bacteria, Archaea, and Eukarya to a level above kingdom, creating the widely accepted three-domain system.
Above kingdom level.
Yeah. And the reason RNA was such a breakthrough is quite elegant, really. It's present in every living cell. Its genes are highly conserved, meaning they evolve very slowly over billions of years.
So they don't change much.
Exactly. And they contain signature sequences that act like unique genetic barcodes for different groups. This allowed scientists to map out evolutionary relationships with, well, unprecedented accuracy.
Genetic barcodes, I like that.
And beyond RNA, these three domains also show fundamental differences in their membrane lipid structures, their transfer RNA molecules, even their sensitivity to antibiotics. If we connect this to the bigger picture, it tells an incredible evolutionary story. These three distinct cell lineages emerged over 3.5 billion years ago.
3.5 billion.
But it wasn't a neat, isolated split. What's truly mind-bending is the concept of horizontal gene transfer.
Right, where genes jump between organisms.
Exactly. Genetic material is shared not just parent to offspring, but between entirely different organisms, blurring those early lines, showing that life has been sharing its genetic secrets for eons. This also feeds into the endosymbiotic theory, explaining how eukaryotic cells, which appeared much later, maybe 2.5 billion years ago, actually evolved from prokaryotic cells living inside one another.
Becoming organelles like
Yeah.
mitochondria.
Precisely, like the mitochondria powering our cells. And the oldest prokaryote fossils, by the way, date back over 3.5 billion years. Truly ancient life.
It really paints an incredible picture of life's interconnectedness.
Yeah.
Okay, now that we have that grand evolutionary framework, let's explore what makes each of these three domains unique. Starting with domain bacteria. These are your classic prokaryotic cells. Their cell walls contain peptidoglycan, that's a unique polymer, right? And a key target for many antibiotics.
That's the one.
Their cell membranes have straight carbon chains linked by something called an ester bond. They kickstart protein synthesis with formylmethionine. They typically have 70 ribosomes and reproduce by simply dividing binary fission. This domain is vast. It includes most common pathogens, but also countless non-pathogenic bacteria in soil, water, our own bodies, even photoautotrophs.
Right. Then moving to domain Archaea, these are also prokaryotic, but with fascinating distinctions that really set them apart. Their cell walls lack peptidoglycan, which means many common antibiotics targeting peptidoglycan just don't affect them.
Ah, okay, so they're resistant in that way.
And their membrane lipids are structurally different. Branched carbon chains, and ether linkage instead of ester. That's a major reason they can thrive in environments where other life forms just can't. Like eukaryotes, they use methionine for protein synthesis. They also have 70S ribosomes, divide by binary fission. But what's truly captivating about Archaea is their ability to flourish in extreme conditions, often called extremophiles.
The extremophiles.
Yeah, we generally group them into three types. Methanogens, which produce methane, strict anaerobes. Extreme halophiles, they demand incredibly high salt concentrations.
Right.
And hyperthermophiles, which just love extreme heat. They really push the boundaries of where life can exist.
Amazing. And finally, we arrive at domain Eukarya, which of course includes us. These are eukaryotic cells, meaning they have a true nucleus, other membrane-bound organelles. Their cell walls, if they have them, are made of carbohydrates, not peptidoglycan. Like bacteria, their membrane lipids have straight carbon chains with ester linkages, and they use methionine for protein synthesis, just like Archaea. They're generally not sensitive to antibiotics, have larger ADS ribosomes, and divide through a more complex process, mitosis. This domain encompasses the familiar kingdoms: fungi yeasts, molds, mushrooms absorbing nutrients, plantae multicellular photoautotrophs, trees, flowers, and Animalia multicellular ingestive heterotrophs, like us. Then there are the protists. Historically a bit of a catch-all group, wasn't it? Simple, mostly single-celled eukaryotes, diverse nutritionally.
Exactly, a real grab bag.
But here's another point of evolution in our understanding. Modern RNA sequencing is now breaking protists down into more genetically related groups, or clades, giving us a much clearer picture of their true evolutionary lineage.
Yeah, tidying up that category significantly. And of course, we have to address the ultimate outsiders, viruses.
Ah, yes, the viruses. Where do they fit?
Well, they don't, really. They defy classification into any of the three domains because they aren't cellular life forms in the traditional sense. They lack ribosomes, lack the machinery to multiply on their own. Instead, they hijack a living host cell's machinery to reproduce.
Right, the ultimate parasites in a way.
You could say that. We define a viral species as a population of viruses sharing similar characteristic shape, genes, enzymes, and occupying a specific ecological niche, usually meaning a particular host cell or tissue. Their exact origin is still a mystery, but theories suggest maybe independently replicating nucleic acids or perhaps degenerative cells, or even coevolution with hosts right from the start. They really represent a unique form of biological entity.
It's clear the microbial world is just incredibly diverse. So with all these fascinating organisms, how do scientists actually keep them all straight? This is where that consistent naming system comes in, right? Scientific nomenclature.
Absolutely vital, yeah.
Imagine the chaos with common names. Spanish moss, you mentioned not a moss, different name everywhere. So scientists use binomial nomenclature, two names: genus and specific epithet, or species, like Homo sapiens for us, or Rhizopus stolonifer for bread mold. Genus capitalized, species lowercase, always italicized or underlined.
Standard practice.
Names are from Latin or Latinized. Specific suffixes for larger groups. Orders end in ales, families in ace. And it's worth noting, reclassification happens. You mentioned Streptococcus faecalis becoming Enterococcus faecalis.
Exactly. That happened after scientists gained deeper understanding from rRNA analysis. It shows taxonomy is dynamic, a living science.
Always updating.
And these names aren't just random. They fit into that structured, nested system, the taxonomic hierarchy, like Russian dolls, you said, each level more specific. Domain, kingdom, phylum, class, order, family, genus, species.
Got it.
Now, when we talk about a eukaryotic species, we generally mean organisms that can interbreed, produce fertile offspring. But for prokaryotic species, it's different. Since they don't reproduce sexually, they're defined as a population of cells with similar characteristics.
Makes sense.
In microbiology, we also use specific terms like culture bacteria grown in lab media, a clone, a pure culture from a single parent cell, ideally genetically identical, and a strain, a pure culture of the same species that shows some slight identifiable differences, often designated by numbers or letters.
Okay, culture, clone, strain. Important distinctions.
Very. And for prokaryote classification, Berge's manual is the definitive go-to guide. It's an enormous resource. It's also fascinating to remember less than 5% of all bacterial species are actually human pathogens. Most are essential to our world.
Only 5%. That puts things in perspective. Okay, so that's a great distinction. Classification tells us about relationships. Identification is more practical, like figuring out what's making someone sick. So once we have a sample, how do scientists actually unmask these microbes in the lab? What are the methods?
Right. That's where the detective work begins. Often combining old and new methods. We start with traditional methods. Morphological characteristics are still useful. While many microbes look like, small rods or spheres, unique features like endospores or flagella can be key clues. Think of Pneumocystis jirovecii, causes pneumonia in immunocompromised patients. For decades, it was misidentified as a protozoan because its morphology was misleading.
Ah, so looks can be deceiving.
Very much so. It wasn't until RNA sequencing in 1988 that we truly understood it was a fungus, which completely changed its treatment.
Wow. Okay. What else in the traditional toolbox?
Differential staining, like the Gram stain, gives quick, vital info about the cell wall chemistry, immediately narrowing down possibilities, gram-positive, gram-negative, and then there are biochemical tests. These detect specific enzymatic activities. Can it break down certain sugars, produce specific waste products? They act like incredibly precise chemical fingerprints.
Like you mentioned with the pneumonia case earlier.
Exactly, and these are often combined into rapid identification systems, test strips or panels that can identify a bacterium within hours.
That's fascinating how those biochemical signatures can be so revealing. What about methods that look at how microbes interact with our immune system?
That leads us to immunological methods, often called serology. The principle is simple but powerful. Microorganisms act as antigens. They trigger our bodies to produce highly specific antibodies.
The lock and key idea.
Precisely. We use the specificity for identification. For instance, in a slide agglutination test, you mix an unknown bacterium with a known antiserum that's a solution with specific antibodies. If you see visible clumping, agglutination, that's a positive ID. This allows us to differentiate not just species, but even serotypes or serovars, distinct strains within a species with unique surface antigens. Rebecca Lansfield's work on strep serotypes is a classic example.
So you can get really specific.
Extremely. More advanced techniques like ELISA enzyme-linked immunosorbent assay are rapid, can be automated. They use known antibodies to detect bacterial antigens or even patient antibodies to a pathogen, like in AIDS testing. And Western blotting takes it a step further. It identifies specific antibodies in patient serum against separated proteins, crucial for confirming infections like HIV or Lyme disease.
These methods sound incredibly precise for identification. But what if you need to track down the source of an outbreak, identifying a specific strain, pinpointing it?
For that, phage typing is an elegant solution. It uses bacteriophages. These are viruses that specifically infect and destroy bacteria.
Viruses that hunt bacteria.
Exactly. And each type of phage is incredibly specialized, targeting only certain species or even particular strains. By observing where these phages cause clearings or plaques on a bacterial lawn, we can identify and differentiate strains. This technique has been instrumental in tracing outbreak origins, linking surgical wound infections to a specific person, or differentiating Salmonella serotypes in foodborne illnesses.
Clever. What about other chemical clues?
Sure, chemical analysis also provides unique signatures, for instance, fatty acid profiles, or FAMI analysis. It looks at the unique fatty acids bacteria produce. These are constant for a species used commercially, and flow cytometry can identify bacteria without culturing them. It detects how cells scatter light or fluoresce, gives info on size, shape, even tagged molecules like detecting listeria in milk.
So we've progressed from looking at the whole organism to its chemistry. Now let's talk about the ultimate level of precision, the molecular level, DNA and RNA. This is really where that third golden age shines, isn't it?
Absolutely. Molecular methods focusing on nucleic acid analysis have indeed revolutionized the field, revealed incredible detail. One of the simpler methods is DNA-based composition, specifically the percentage of guanine plus cytosine, the G plus C content. Closely related organisms have similar G plus C percentages. A difference over, say, 10% strongly suggests they're unrelated.
A quick check for relatedness.
Kind of, but for true precision, DNA sequencing comparing entire base sequences is the gold standard. This led to discovering new pathogens like Borrelia miyoni, a new Lyme cause, crucial for microbes without fossil records.
Right. And then there's fingerprinting.
DNA fingerprinting, yeah. Uses restriction enzymes to cut DNA into unique fragments called RFLPs. Separated by electrophoresis, they create distinct patterns. More similar patterns mean more closely related. This has been invaluable for tracing hospital-acquired infections, pinpointing outbreak sources like connecting patient infections of Gordonia bronchialis back to a specific healthcare worker.
Hospital detective work.
Pretty much. And nucleic acid amplification tests, NATs, especially PCR-based methods, are game changers. They amplify tiny amounts of microbial DNA or RNA from a sample. It makes it possible to detect organisms impossible to culture.
Like the Whipple's disease microbe. Or Zika.
Exactly. Trophima whippleau, Zika virus, ancient DNA sometimes. But perhaps the most powerful tool for understanding evolutionary relationships among microbes is ribosomal RNA sequencing, especially the 16S RNA gene.
Why the 16S gene specifically?
Well, a few reasons. All cells have ribosomes. The 16S RNA gene changes very slowly over evolutionary time. And crucially, you don't even need to culture the organism to sequence its RNA.
Ah, so you can analyze environmental samples directly.
Precisely. This led to incredible discoveries like Pelagibacter, now known as the most abundant organism in the ocean. Didn't know it existed before we could sequence environmental DNA. And finally, fluorescent in situ hybridization, uses dye-labeled RNA or DNA probes to directly stain microorganisms in situ right in their natural environment, to identify them, count them, even assess their metabolic activity. Staph aureus in a patient, bacteria in drinking water.
Wow, that's an incredible arsenal of techniques, such detailed insights. So how do all these pieces from basic taxonomy to cutting-edge molecular methods fit together in, say, a real-world scenario? Let's trace that hypothetical salmonella outbreak. Monica Jackson, her friend, diarrhea after a luncheon.
Okay, yeah. This is a perfect example of public health in action. Multiple methods layered together. First, the lab gets Monica's stool sample. Instead of just a gram stain, which would show tons of indistinguishable gram-negative rods, they culture it on specialized selective and differential media. These inhibit many common bacteria, let salmonella grow, and make it show distinct characteristics.
Okay, so you isolate it first.
Right. This quickly identifies salmonella enterica, but that's just step one. There are over 2,500 serovars of S. enterica.
2,500.
Yeah. So the health department then performs serotyping to identify the specific serovar, maybe Salmonella tennessee in this case. Then, when more cases start popping up across different states, same serovar, the CDC steps in with DNA fingerprinting.
Right, the RFLPs.
Exactly. They take DNA from the salmonella, isolates from all the infected people, compare their unique RFLP patterns. This allows them to link those, say, 29 cases to a nationwide outbreak. They can even differentiate strains, maybe rule out an earlier cluster linked to raw cookie dough as distinct from this outbreak, which was eventually traced to hydrolyzed vegetable protein in snacks. Yeah. This comprehensive, multi-layered approach is how public health protects us every day.
It's truly amazing how they can track something so tiny across an entire country. And it's not just about human health, is it? These techniques apply elsewhere.
Oh, far from it. Consider veterinary microbiology. We've seen devastating mass deaths of marine mammals, dolphins succumbing to brucella, sea otters declining due to toxoplasma. Here, conventional tests, genomic data, FIH, all vital tools to identify these pathogens. This work impacts wildlife management, ecosystem health, even provides models for human diseases. And perhaps one of the most exciting new frontiers is exploring the human microbiome. The incredibly complex communities of microbes living on and in us. Historically, we had to culture them, but we now know many just can't be grown in the lab.
The unculturables?
Exactly. New DNA analysis, particularly sequencing that 16S rRNA gene, lets us identify and understand relatedness without culturing. Whole genome sequencing gives the most complete picture, but 16S is a fantastic starting point for mapping phylogenetic relationships. But it's crucial to remember old and new techniques are both essential. They complement each other. Genetic analysis tells us what's there, but culturing helps understand what they do. Their metabolism, host relationships, discovering things like new antibiotic resistance genes in action.
A really integrated approach then. So from ancient fossils whispering stories of early life to cutting-edge DNA chips that can rapidly diagnose disease, you've now taken a deep dive into the incredible science of classifying and identifying microorganisms. It's a field that's constantly evolving, just like the microbes themselves. And it's truly fundamental to understanding everything from, well, human health to the intricate workings of global ecosystems.
What's truly fascinating here, I think, is how our understanding of microbial relationships, from basic taxonomy to detailed genetic fingerprints, isn't just about labels on a diagram. It's about revealing the hidden stories of adaptation, survival, constantly pushing the boundaries of what we thought possible in this world, teeming with invisible life.
Indeed, and consider how that constant coevolution between microbes and their hosts drives not only the immense diversity we classify, but also shapes the very tools we need to understand them. What surprising new classifications might emerge as our understanding of horizontal gene transfer deepens, blurring even more of those long-held evolutionary lines. Makes you think. Thank you for joining us on this deep dive. Until next time, stay curious.